diversity and environmental preference via functional similarity networks.
Yeah, thank you, Ido. It is a big pleasure to be here, and I'm super excited to tell all of you about fusionDB. As Ido already said, we are able to assess microbial diversity and environmental preference via functional similarity networks with this tool. So we pretty much have two fundamental questions in microbiology. First of all, who is present? And second of all, what can they do? The first question can be answered quite easily with an array of established methods that are out there that are especially good at, for example, assessing the environmental, the evolutionary relatedness between samples or between specific microbes. However, there is one problem, and that is evolutionary relatedness cannot guarantee functional relatedness. To name one specific example, your standard E. coli lab strain is about 40% smaller in terms of its genomic size than the E. coli that is from time to time responsible for infections and hospitalization of people. So as you might have realized, and that's why I am here, we are actually much more interested in what those microbes can do. And for that, we came up with a concept called fusion. We are comparing microbes based on their functional relatedness. So the project started out with a selection of about 1,400 organisms that were at the time when the project started available in the public databases, microbes that were fully sequenced. That translates to about 4.2 million proteins. We take those 4.2 million proteins and dump them into our fusion machine. And what we ultimately want to end up with is we want to know whether two proteins share the same function, because that's important for our next steps. So what we do is we take an all-against-all SciBlast, and we are specifically interested in two values, namely the sequence identity and the ungapped alignment length. Because what we are doing is we're taking HSSP, which is a measure that can help us assess whether two proteins are of the same function. So what HSSP does is it takes a pair of proteins and places it into a space that is defined by sequence identity and alignment length. And depending on where this protein pair falls, either above the curve or below the curve, it will tell us whether the function is the same or different. It is important to note here that the greater the distance of the protein pair from the curve in the vertical, the more significant the actual functional similarity is. So we're ending up with an array of HSSP scores for our predictions, and we are only interested in those where the HSSP score is bigger than 10. Those protein pairs that are fulfilling this criterion will then be connected in a similarity graph via edges. And this similarity graph we are running through Markov clustering. Markov clustering enables us to dissect the graph and pretty much put the proteins into functional clusters. So you might remember that I said in the beginning that we started out with 4.2 million proteins, and what we're actually ending up with is around 460,000 functional clusters. And I want to stress here that only about a third of those are actually clusters that contain multiple proteins. So 300,000 out of those 460,000 protein functional clusters are actually protein, respectively functional unique. Having those functional clusters, we're then backtracing and define our microbial repertoire. So the specific example here, in functional cluster 1, we have protein 1, protein 4, and 5. Those proteins were originating from microbe 1. So since in our functional cluster 1, we have proteins from that specific organism, we're going to assign that functional cluster to the repertoire of the first microbe. And having established those functional repertoires, we can then finally get to our functional relatedness. The functional relatedness is here defined as the size of the intersection between two repertoires, so which functions they share, divided by the bigger functional repertoire. In the example of the repertoire for microbe 2 and 3, function 1 would be the one that is shared between the two microbes, and then we're dividing it by the size of the repertoire for microbe 2. Having those relations established, we can then visually represent the actual interactions. So we're offering two visual representations. One we're calling Fusion, the other Fusion Plus. Fusion is a complete graph where each of the organism nodes are connected with all of the other nodes. The thickness of the edge, respectively, the weight of the edge is here defined by the actual functional relatedness between those microbes. Into Fusion Plus, we're introducing what we're calling function nodes. It is important here that the organism nodes are only indirectly connected to each other. So there is no direct connection between organism nodes. They are connected via all of the functions that they share. So why am I telling you all of this? We actually started looking into instances of potential horizontal gene transfer. So what we did in this specific example is we extracted a number of microbes from the taxonomic class of Clostridia and a number of bacteria from the taxonomic class of bacilli. And what you can see here is if we're coloring this network by their temperature preference, there is a very clear separation according to the environment that they're actually present in. However, if we're going back to the taxonomic separation, things are not as clear anymore. Specifically, if you're looking at the by the star-shaped outline organism, this one is a bacilli that clearly clusters into the Clostridia microbes. And yes, we have 3 thermophilic bacilli in there. And the point why I'm telling you this is, if we're then looking into the taxonomic tree, there again is a clear separation between the taxa, for obvious reasons. But if we're looking at pyruvate phosphate dekinases, and we're building a phylogenetic tree based only on this protein, the 3 thermophilic bacilli actually cluster much closer to the Clostridia than the rest of the bacilli. And this might actually be an indication that the specific occurrence of that gene in those 3 bacilli might have been horizontally transferred from the Clostridia. So you might ask, where does my organism fit? So if you come in with a new organism, as I said in the beginning, we started building the database with 1,400 organisms. By now, there are much more organisms in the publicly available databases present. So we are currently working on updating the database, but in the meantime, we wanted to give you the opportunity to actually investigate your own microbes. So for the sake of it being, just going to call it Steve. So where does Steve fit into our functional network? We started by using the microbes that we already have as our reference database, and we're essentially running the Steve against the database through SciBlast. We're running it through HSSP again, and then take only the highest scoring hit according to HSSP. Knowing to which protein in the reference database it maps, we then can trace back and take out all of the functions, and with that recreate a functional repertoire of Steve. I want to point out here that we evaluated a couple of more methods how we can actually do the mapping, and ended up using just taking the highest scoring pair, because it actually shows a very high performance of around 90% precision and recall in terms of recreating the actual functional repertoire. So the mapping of Steve, I'm actually going to show you in a small example. So if you're navigating to our web service, you have either the option to explore what is there or look where Steve fits in. And there you're faced with two options. Either you're pasting your proteome into the web mask, or you can upload the proteome into the pipeline and then run everything through it. As soon as you have submitted your job, you will get updated with a status page that will detail a little bit about how far the job has progressed. And as soon as the job is done, you will be welcomed with our result page that details which functional clusters are actually present in the functional repertoire. But more importantly, we are also listing all of the organisms that are present in our database with the actual functional relatedness value. You can search for whichever organisms you're interested in. In this specific case, Steve was actually a Cyanococcus organism that was extracted from a bog. So we're taking all of the Cyanococcus species that we have in our database and then create a visual representation of it. The visual representation, creating this visual representation might take a small moment, unfortunately, because we are in this specific example actually creating and layouting and rendering a network with more than 20,000 organisms. So this is a small, intensive task. Having the standard representation, you are then welcome to recolor the network with your preference. So in this specific example, we looked for a coloring by habitat. And what I want to stress here and point out to you is that, yet again, you can see a clear separation according to the environmental preference, with one exception. And this is, if you look on the top side of the screen, there seems to be one microbe clustered into the freshwater organisms that is actually a marine organism. And what we can do now is we can actually go and explore this graph interactively and get a little bit more information on the actual organism nodes. So we can look for all of the metadata that is available in our database. And if we're actually then going to zoom in into this one organism that is standing out here and look at the actual representation, which will happen right about now, then we will actually see, okay, this organism is extracted from not only marine environment, but actually from a mud environment. And we use this sort of as a starting point to go back into the literature and research a little bit more, and figured out, okay, so yes, this organism is coming from a marine environment, but in fact, despite it being salt tolerant, it does not require salt to grow. So it will grow equally well in freshwater environment. And as you saw from the representation, it might actually cluster functional-wise much closer to the rest of the freshwater environment organisms than to the marine organisms. Just a small finishing picture. So you also have the option of downloading the whole network and then choose your visualizer of choice, in this specific example, GEFI, and then adapt the representation to your liking, make it a little bit more pretty. So in conclusion, what I want to point out here, the functional similarity is more representative of environmental requirements than evolutionary heritage. We also enable you to map previously undescribed organisms to their likely environmental preference, and most important, of course, use the tool. And with that, I want to thank all of the people of the Bromberg Lab, especially Jana, Shen Zhu, and Max Miller that helped me creating all of this, and also the NSF and European Commission for funding this project, as well as the NSF for the actual travel fund.
Okay. Any questions? Yes.
Hi, thank you for the talk. The question is, am I able to get data from your database for passing it through my own pipeline later on, or should I do it only on your site?
Well, we would obviously welcome it if you would do it on our site, but there are also data sets available that you can download.
Okay, but I mean, kind of, I have some experimental data. I will find the results on your database, and I would like to kind of download them and analyze later on with my own
You can download
results of analysis.
results from that database, yes.
Thank you very much.
I have a question with respect to the resolution of function that you have. So you are talking about two types of functions. One was the molecular function where you show HSSP curve where individual pairs of proteins interact. And even you may not know the molecular function, you're still able to kind of map it later to the environmental, let's say, features that are provided. But my question is, sometimes it could be one gene that changes things. I guess my feeling is, what is your sense of sensitivity of your method? If you're mapping it to a broad category, you're expecting that one gene to influence the whole organism to move from one part of the graph to kind of be clustered with the other part of the graph. And while I understand that you have success stories, do you have a sense how often those success stories occur? And
we have actually a couple of more examples, yes. So, of course, the shared functions that are very broad will only occur in the middle. But the point that we want to make is that the individual functions that might, yes, be only organism-specific functions are still equally important to distinguish special functionality of organisms. So we have an example of mycoplasma bacteria, and fusion as well shows that like it pulls out a couple of mycoplasma that are only present in blood and very parasitic, and completely separates it from all of the other mycoplasma that are in our database. So my answer to that would be, we think that those individual functions are equally important to actually be able to distinguish environmental preferences as the core functionome, so to speak, yes.
That's a lot to ask for algorithms to do that.
Hi, very nice talk. I have one quick question. If because you classify organism based on the predicted function, there will be some sequences which you cannot give function annotation. So compared to the function-based clustering with more like a conventional sequence-based clustering, which can cover all the genomes, what else? What is the advantage of using your function-based clustering?
So to very briefly go to the functional assessment. So there are actually most of the functional clusters that are available in the database have only hypothetical annotations or actually don't have any annotation. But that sort of goes back to the previous question. Even if we do not know the function itself, we still think that is vital in separating the organisms out. And I would argue that placing those organisms into an environmental understanding is actually one of the main goals of all of this.
Hi, thank you. Did you try to use these functional traits to also describe antibiotic resistance patterns? Would it be possible to know when you have more complex systems, efflux pump function or stuff like that?
We actually did not do that yet, no.
So I want to follow up a little bit on that question. You're looking at horizontal gene transfer, which looks like horizontal gene transfer on chromosomal levels that has taken place evolutionary, a while ago. And a while can be, 5 years or 10 years. I mean, these are microbes after all. Following up on the ABR question, I want to ask something more general, because in the biomedical community, the interest would be horizontal gene transfer that took place 3 months ago or 5 months ago in form of antibiotic, any kind of pathogenicity. It does not have to be antibiotic resistance, it can be anything, but ABR is a big thing. And that takes place actually on plasmids, usually, with pathogenicity islands, not on chromosome. Do you have any sense of whether you can tackle that with your method?
So the current data set that we have in FusionDB and the genomes that were used to build the whole relationship did not include plasmids at that point. We are, while we're updating the database, including plasmids, and we will probably figure exactly this question out sometime soon.
Okay, I'm sorry, just one more question. I'm sorry, Luis.
So very quickly, for the similarity of function, the way you define that two proteins should might retain the same function, I'm kind of surprised that you are not using orthologs to try to define that. I suppose that it's for computational time. But did you try to apply the same kind of method with as input orthologs sequences that you can have in some public databases like OMA or Ortho? And do you find the same result in that case?
We did not do this.
Okay, thanks.
Okay, thank you. We need to move on, so we can thank our speaker once more.